Greetings to the learners. Today's topic to learn is repetitive DNA. Repetitive sequences refers to DNA fragments that are present in multiple copies in the genome. Mammals have approximately 3 billion base pairs per haploid genome, harboring about 20,000 to 25,000 genes. A minor part of the genome, 5 to 10%, is coding sequences, and the remaining is non-coding, representing the repetitive DNA. Comparison of the genome size of different eukaryotes shows that the amount of non-coding DNA is highly variable and constitutes 30 to 99% of total genome. The non-coding repetitive sequences are dynamic elements which reshape their host's genome by generating rearrangements, shuffling of genes, and modulating pattern of expression. This repetitive DNA can be categorized as highly repetitive and moderately repetitive. Highly repetitive contains satellite DNA and moderately repetitive has tandem repeats and interspersed retrotransposons. Tandem repeats includes multiple copy genes, mini satellites and microsatellites, whereas interspersed retrotransposon contains signs and lines. Signs is short interspread nuclear element and lines is long interspread nuclear element. These highly repeated DNA sequences are also known as reiterated or redundant DNA. They account for nearly 10% of the total DNA in vertebrate genomes. They are very short and usually ranging from few hundred base pairs, and they are arranged in tandem arrays in an end-to-end pattern. These highly repetitive sequences are usually found in specific heterochromatic regions such as centromeres, pericentromeres, and telomeres. They also play roles in chromatin organization and genome evolution. Types and examples of highly repeated DNA. Satellite DNA, mini satellite DNA, and microsatellite DNA. Satellite DNA are very short nucleotide sequences typically ranging from 5 to 100 base pairs, and they are highly species specific, and many organisms possesses multiple satellite families. These sequences are often localized in heterochromatic regions such as centromeres and telomeres, and they contribute to chromosome structure, segregation, and stability. Biologically, satellite DNA plays important roles in maintaining centromere function, ensuring proper kinetochore attachment, regulating chromatin organization, and provides species-specific genomic signatures. Mini satellite DNAs. They consist of moderately about 3000 repeat units, and they range from 12 to 100 base pairs in length. Compared to classical satellite DNA, mini satellites occupy smaller and more localized genomic regions, yet they are still present in significant numbers throughout the genome. One of the most striking features of mini satellite DNA is its high level of instability. They are commonly found near telomeres, subtelomeric regions, and other regulatory areas of chromosomes. Their instability can arise from processes such as unequal crossing over, slipped strand misrepairing, or gene conversion. Microsatellite DNA, which is also known as short tandem repeats or simple sequence repeats, consist of very short nucleotide motifs repeated in tandem throughout the genome. Their length ranges from 1 to 6 base pairs. These microsatellite sequences are widely distributed across both coding and non-coding regions of the genome, though they are particularly abundant in introns, intergenic regions, and regulatory sequences. Microsatellites are extremely mutable and unstable, primarily due to a mechanism known as replication slippage during DNA replication. When the DNA polymerase slips or mispairs, the number of repeat units can increase or decrease, leading to frequent expansions or contractions of the microsatellite region. This variability results in high polymorphism, where individuals differ significantly in the number of repeat copies at a given locus. Lines. Long interspersed nuclear elements are found throughout eukaryotic genomes. They are typically long, autonomous transposable elements that range from 6,000 to 7,000 base pairs in length. And lines are scattered across the genome at thousands of distinct locations rather than grouped in clusters. Looking into the structure of these lines, most of the lines contain two open reading frames, that is, ORF1 encodes an RNA binding protein, ORF2 which encodes a protein with reverse transcriptase and endonuclease activities. A 5' untranslated region that often includes an internal promoter, and a 3' untranslated region that usually ends with a poly A tail. Autonomous mobility. Lines are considered autonomous retrotransposons because they can transcribe themselves into RNA, use their own reverse transcriptase to convert RNA back into DNA, and insert the DNA into the genome at new sites. Their method of movement is called target primed reverse transcription. Signs. Signs are short interspersed nuclear elements. They are a category of moderately repeated non-coding DNA sequences that are widely distributed throughout the eukaryotic genome. They are typically 100 to 400 base pairs long, making them much more shorter than lines. Although signs themselves do not encode any proteins, they are mobile genetic elements that propagate through the genome using a mechanism called retrotransposition. Looking into the characteristics of signs, they are short in length, high copy in number, non-autonomous transposon, and their origin from small cellular RNAs, particularly tRNA, 7-SL-RNA, and 5-srRNA. Transposons or retrotransposons. Transposons are also called as the jumping genes which move from one location to another within the genome. They are broadly classified into DNA transposons and retrotransposons based on their mechanism of mobility. DNA transposons move through DNA intermediate. They encode an enzyme called transposase, which cuts the transposon out from its original site and inserts it into a new genomic location, and this process is called as cut-paste mechanism. Their structure contains transposase gene, terminal inverted repeats at both the ends, target site duplication, flanking the insertion site. Retrotransposons. Retrotransposons move through an RNA intermediate and follow a copy-paste mechanism. Transposon is transcribed into RNA. It's converted into a complementary DNA by the enzyme reverse transcriptase, and the cDNA gets inserted into the new genomic location. Because of the original copy remains in place, retrotransposon increases the genome size significantly. There are two types of retrotransposons. One is LTR and the other one is non-LTR retrotransposons. LTR, they possess long terminal repeats at both the ends, and they contain genes for reverse transcriptase, integrase, GAG-like proteins. And examples, TY elements in yeast, Copia and Gypsy elements in Drosophila, ERVs, that is endogenous retroviruses in mammals. Non-LTR retrotransposons, they lack long terminal repeats and they include lines and signs. Thank you all.